Hello and welcome to the Penguin Prof channel. In today's episode, I want to talk about the cell cycle and mitosis. Don't be too afraid. I know this looks pretty ugly, but of course we're going to have some fun with it, and I'm going to introduce you to all the players that are involved and hopefully make this pretty easy to understand. And of course there's cleavage. So, what else do you want? Have you ever felt, lonely? Have you ever thought to yourself, gosh, there's really no one I can talk to that understands me? What if you could clone yourself? I know for some that's a scary thought, but if you're a single-cell organism or a colonial organism, cell division actually allows you to do that. For these guys, it's all about reproduction. And cell division is important for single-cell organisms, for colonial organisms like this coral. They can grow asexually by cloning themselves. They can also reproduce sexually where they shuffle their genes. But this basic cell division we're going to be talking about today is important for them. It's also important for us because this is the kind of division that allows for the development of a single fertilized egg into a baby. And of course, it allows for you to replace cells that get worn out or damaged. So cell division is really important. But there are a lot of challenges and problems that we have to deal with. You have to make sure that when one cell divides into two, the daughter cells, they're just called daughter cells by convention, they must be identical. And the management of DNA is key. We're talking about a lot of DNA. For humans, we're talking about 6 billion base pairs with about 50,000 protein encoding genes. This is a lot of DNA. If you stretched it out, there would be 6 feet of it. How are you going to manage all of that? If you couldn't imagine that, I went ahead and got 6 feet of string, and it's kind of a mess. But if you coil it up, it's actually a lot easier to manage. And this is actually what the cells do with their DNA. So when the cell isn't reproducing its DNA or dividing, it exists in a loose, kind of uncoiled formation called chromatin. But when the cell is going to be dividing, all of that DNA gets organized into big groups called chromosomes. So these are what human chromosomes look like. When we sort them out by size, we call this a karyotype. And you can see how the cell organizes all of the genomic material so that it's easier to manage during the process of cell division. It's critical that we manage it because if you lose even a tiny part of a chromosome, the results can be catastrophic and can lead to disease or death to the cell or the organism. So this is really serious business. So the cell cycle is going to involve this kind of an overview. We're going to have a parent cell with the genetic material shown in red and blue. There's going to be a stage where the DNA is going to be replicated. And then you're going to see the classic X shape of the chromosomes. And then there's a distribution phase. So you're going to have half of each of the X's being moved to opposite sides of the cell, and then the cell is going to divide in two. So in terms of the big picture, that's what's going to have to happen. Now, there's a huge misconception alert here. A lot of people think that the replication of DNA is part of mitosis, and it is not. You have to duplicate the DNA before mitosis begins. So we're going to see here in a second at what phase the DNA is actually replicated, but it is not during mitosis. Okay, so we're going to talk about the life of a cell. And my mentor, Todd Newberry, said that organisms organize space with structures and time with life cycles. And so I want to share with you the life cycle of a cell. You can basically divide the cell's life cycle into two periods: when the cell is not dividing and when it is dividing. And actually the cell spends most of its time not dividing. This non-dividing phase we call interphase. Inter means between. And the cell division stage we refer to as cell division. And of course we're going to subdivide these into more phases because that's going to allow us to look at different events that occur. So interphase is divided into 3 subphases: the gap 1, the synthesis, and the gap 2 phase, and we'll talk briefly about what happens there. And then the cell division phase is actually subdivided into two phases: mitosis, which is the division of the genetic material, the DNA, and then the cytokinesis, which is the division of everything else. So the cytoplasm and the actual creation of two cells occurs during cytokinesis. So during gap 1, this is when the cell has just finished division. So it's generally very small in size, and it has a lot of growing to do. It has a lot of needs in terms of metabolism and some preparation for DNA synthesis. The S phase, the synthesis phase, is when the DNA gets replicated, and that is also when the centrosomes, these are organelles that we're going to see in a few minutes, they play a critical role in cell division. They get replicated as well. So DNA is not replicated during mitosis, it is replicated during the synthesis phase of interphase. So that's your big misconception alert there. The gap 2 is going to be more growth, more synthesis of proteins, a lot of synthesis of things that are going to be needed in order to go through the cell division process. So a lot of microtubules, as you're going to see in just a bit. And those are the phases of interphase. Now, a little bit more about chromosome structure is going to be very helpful at this point. These chromosomes are always drawn like X's, and they actually do look like this. This is the real structure. So it's an X, and there's a centromere at the center. If you were to divide the chromosome in half, what you would see is that each half is a replicated sister of the other half.